Hello all. Today's topic is about the history and the scope of microbiology. So I already uploaded a video on introduction to microbiology. So in this video we see about the introduction and important events in the development of microbiology and its scope. So microbiology is nothing but the study of microorganisms. So the microorganisms which include bacteria, algae, fungi, slime molds, viruses and protozoa. So bacteria is a prokaryotic organism. Algae, it is a photosynthetic protist. Fungi, it is a diverse group of organisms and it plays a both beneficial role and it also causes a plant disease and animal disease. So coming to the slime molds, it both like a protozoa and like fungi. So like protozoa in one stage of life cycle and in other stage it's like a fungi. So then viruses, it is the smallest of all microbes and for example smallpox, rabies, influenza, etc. Then protozoa, it is a unicellular animal-like protist and it is a motile organism. So these microbes which play a key roles in nutrient cycling, biodegradation, climate change, food spoilage, cause and control of disease and biotechnology. So next coming to the history of microbiology, various events in the development of microbiology. So in the year 1546, Castro suggests that the invisible organism which causes a disease. In 1590 to 1608, Janssen develops the first useful compound microscope, and in 1665, Hooke publishes Micrographia. That is the first drawing of a microorganism which published in the Micrographia. In 1676, Anton von Leeuwenhoek discovers the animalcules. He was the first person to observe the microorganisms with the help of the simple microscope. And in the simple microscope, organism appear as a bright object against a dark background, and he named it as an animalcules. In 1688, Reddy refuses a spontaneous generation of maggots. In early times, people had believed in the spontaneous generation theory, but the Italian physician Francisco Reddy, he conducted the series of experiments on the decaying meat and its ability to produce the maggots spontaneously. With the help of the experiment, he concluded that the generation of maggots by the decaying meat results from the presence of the fly eggs only, and the meat did not spontaneously generate the maggots, so he refuses the spontaneous generation theory. Then in 1765 to 1776, Spallanzani attacked the spontaneous generation. In 1786, Miller produces the first classification of bacteria. In 1798, Jenner introduces the cowpox vaccination for smallpox. Edward Jenner used the material from the cowpox lesion to protect the people against the smallpox disease. In 1838 to 1839, Swan and Scladden proposed the cell theory. In 1835 to 1844, Bassi discovers the silkworm disease caused by the fungus. Agostino Bassi first showed a microorganism which could cause the disease, that is, the silkworm disease is due to the fungal infection. In 1847 to 1850, Semmelweis introduces antiseptics to prevent the disease. And in 1857, Louis Pasteur described the fermentation. In 1861, Louis Pasteur disproved the spontaneous generation theory using the Smaragd flask. In 1867, Lister publishes on the antiseptic surgery. Joseph Lister developed the antiseptic surgery system to prevent the microorganism from entering the wounds. In 1876 to 77, Robert Koch, he is a German physician, demonstrated the anthrax disease which is caused by the Bacillus anthracis. In 1880, the scientist Laveran discovers Plasmodium, which is responsible for the cause of malarial disease. And in 1881, Robert Koch cultures the bacterium uses gelatin, and Louis Pasteur develops the anthrax vaccine for anthrax disease. In 1882, Robert Koch discovered the Mycobacterium tuberculosis, and for this discovery, he was awarded the Nobel Prize in Physiology or Medicine in 1905 for his work. In 1884, Robert Koch postulate published, and L. Michnikov described the phagocytosis process, and autoclave developed, and Gramstein technique was developed. In 1885, Louis Pasteur developed the rabies vaccine. Escherichia discovered the E. coli bacteria. In 1887, Richard Julius Petri develops the Petri dish. In 1887 to 1890, Russian microbiologist Sergei Vinogradsky studied the sulfur and nitrifying bacteria and so many other contributions in the soil microbiology. In 1889, Bejernik isolated the root nodule bacteria. Martinus Bejernik isolated the aerobic nitrogen fixing bacteria, Acetobacter. In 1890, von Begring's antitoxin for diphtheria and tetanus. In 1894, Kitasato and Elson discovered the Yersinia pestis, which is responsible for the cause of plague disease. And in 1895, Jules Bordet, he is a Belgian immunologist and microbiologist, discovered the complement. In 1896, Van Ermengem discovers the Clostridium botulinum, and which is responsible for the cause of botulism disease. In 1899, Dmitry Ivanovsky and Bejernik uses the Chamberlain filter to study the tobacco mosaic disease, and the Bejernik proves virus causes the tobacco mosaic disease. So in the year 1900, Reed proves yellow fever transmitted by the mosquito, and in 1902, Landsteiner discovered the blood groups like AB, and O blood group. In 1903, Wright and others discovered the antibodies. 1905, Scordian and Goffman showed that Treponema pallidum causes this syphilis disease. In 1906, Wasserman develops the complement fixation test for the syphilis disease. In 1910, Rickett shows a Rocky Mountain spotted fever caused by a microbe. In 1911, Rouse discovers a virus which can cause the cancer. In 1915 to 1970, De Herel and Duart discover the bacterial viruses. 1921, Fleming discovers the lysozyme, and in 1923, the first edition of the Burgess Manual. In 1928, Griffith discovered the bacterial transformation. In 1929, Alexander Fleming discovers the penicillin. In 1931, Van Neyl studies the photosynthetic bacteria. Like that, in various years, scientists' name and their discovery and development in the field of microbiology is mentioned. So, till 2005. So, next, coming to the scope of microbiology. So, these are the various branches of microbiology. So, first one, medical microbiology, which deals with the diseases of humans and animals. And next one, public health microbiology. It is closely related to the medical microbiology. Next one, immunology. It is concerned with how the immune system protects the body from the pathogens and the response of the infectious agent. Next one, agricultural microbiology, which deals with the impact of microorganisms on the agriculture. Next, microbial ecology. It studies the relationship between the microorganism and the components of the living and non-living habitat. In food and dairy microbiology, the use of microorganisms to make the food such as cheese, yogurt, etc. In industrial microbiology, the use of microorganisms to make the product like antibiotic, vaccines, etc. So in microbial physiology, the microbial growth, the study of microbial growth and their metabolism. And in microbial genetics and molecular biology, focuses on the nature of the genetic information and the use of the microorganisms to understand the gene structure and function. That's all about the scope of microbiology.